Assalamualaikum warahmatullahi wabarakatuh. This video is all about our main excretory organ, kidney. Each human body contains two kidneys, which excrete metabolic waste products, toxic substances, and foreign bodies from our human body. The two kidneys situated on the posterior abdominal wall on each side of the vertebral column outside the peritoneal cavity. The right kidney is slightly lower than the left one. The two kidneys form urine from excretable materials carried in the blood. From kidneys, urine is conveyed to the bladder through two ureters. At last, urine is excreted to the external environment by urethra. Each kidney is surrounded by a tough fibrous capsule. Now, let's talk about macroscopic structure of kidney, which has outer renal substance and inner renal sinus. Outer renal substance includes outer cortex and inner medulla. Renal medulla presents about 8 to 10 cone-shaped renal pyramids. Each pyramid presents a base directed to the cortex and an apex projected into the renal sinus as renal papilla. Each papilla is perforated by 16 to 20 or more ducts of Bellini and received by a minor calyx. The pelvis divides into 2 to 3 major calyxes, and these in turn divide into 7 to 13 minor calyxes. And renal cortex consists of renal columns and cortical arches. Renal columns extend to the renal sinus between the adjacent renal pyramids. Cortical arches intervene between the bases of the pyramids and the surface of the kidney. Further, renal pelvis is continued as ureter. Here comes microscopic structure of kidney. Each kidney is composed of 1 to 3 million closely packed tortuous uriniferous tubules. Each tubule consists of two distinct parts. One is excretory part and another one is collecting part. Excretory part includes nephrons, which are the structural and functional units of kidney. Each nephron consists of two parts: renal corpuscle for filtration of substances from the plasma, and renal tubule for selective absorption of substances from glomerular filtrate. Renal corpuscle made up of glomerulus and Bowman's capsule. Renal tubule made up of proximal convoluted tubule, loop of Henle, and descending and ascending limbs of loop of Henle, distal convoluted tubule, and junctional tubules. Whereas collecting part includes collecting tubules into which nephrons open, and ducts of Bellini formed by many collecting tubules. They open into the minor calyxes into summit of renal papilla. Each nephron contains a tuft of glomerular capillaries called the glomerulus, through which large amounts of fluid are filtrated from the blood, and a long tubule into which the filtrated fluid is converted into urine on its way to the pelvis of the kidney. The total glomerulus is encased in Bowman's capsule. Fluid filtrated from the glomerular capillaries flows into Bowman's capsule and then into the proximal tubule, which lies in the cortex of the kidney. From proximal convoluted tubule, fluid flows into the loop of Henle, which dips into the renal medulla. Each loop consists of a descending and an ascending limb. From loop of Henle, fluid enters into the distal convoluted tubule, which, like the proximal tubule, lies in the renal cortex. Distal tubule is followed by the connecting tubule and the cortical collecting tubule, which lead to cortical collecting duct. After that, cortical collecting duct runs downward into the medulla and becomes the medullary collecting duct. In each kidney, there are about 250 of the very large collecting ducts, each of which collects urine from about 4,000 nephrons. Lining epithelium of glomerular capillary, which has outer visceral layer and inner parietal layer. Outer visceral layer is lined by a specialized epithelium which is called podocytes. The podocytes have numerous pseudopodia that interdigitate to form filtration slits along the capillary wall. And outer parietal layer is lined by simple squamous epithelium. Proximal convoluted tubule is lined by simple cuboidal epithelium with microvilli, and distal convoluted tubule is also lined by simple cuboidal epithelium. After that, thin descending limb of loop of Henle is lined by simple cuboidal to simple squamous epithelium, and again ascending limb of loop of Henle is lined by simple cuboidal epithelium. At the end of the thick ascending limb is a short segment that has in its wall a plaque of specialized epithelial cells, which is known as macula densa. Macula densa plays an important role in controlling nephron function. There are three types of specialized cells which are collectively known as juxtaglomerular apparatus. These are juxtaglomerular cells, macula densa, and Lasik cells. Juxtaglomerular cells are located on the afferent arteriole and efferent arteriole. Lasik cell founds at the junction of outer layer of afferent and efferent arterioles. This juxtaglomerular apparatus plays an important role in regulation of blood pressure, autoregulation of glomerular filtration rate. These vascular structures supplying the juxtamedullary nephrons also differ from those supplying the cortical nephrons. For juxtamedullary nephrons, long efferent arterioles extend from the glomeruli down into the outer medulla and then divide into specialized peritubular capillaries called Vasa recta, which plays an essential role in the formation of concentrated urine.